Maraviroc, here's its structure here. And as you can recall, on each of these slides, I will also continue to paste different parts of the life cycle. So if you pay attention to the upper right-hand corner on each of the slides about each class of the drugs, it'll show you kind of where it works. And referring back to that original photo on the previous slide. So here is Maraviroc. It's a entry inhibitor. So how it works is it binds the CCR5 receptor, which is this here. The CCR5 receptor is a receptor of the CD4 cell. So it's this receptor essentially right here in this photo. A fun fact, it is also the only approved antiretroviral drug that targets a host protein. So that's a cool fun fact. It prevents HIV GP120, so one of those envelope glycoproteins that we talked about. And it prevents that GP120 receptor from binding and entering the host cell. So it basically blocks this part right here. So once it does bind, it causes a conformational change in the co-receptor here that prevents the actual HIV GP120 receptor from binding with the CCR5 co-receptor. So just a simple way of saying that is it'll bind here, it causes a change to the GP120, and therefore this interaction can no longer happen. So appropriate use of CCR5 antagonists or CCI entry inhibitors depends on the knowledge of the patient's HIV subtype. So actually, in order to use an entry inhibitor, we do require a testing, and it's actually called a tropic or tropism testing. That helps to determine the HIV subtype because there are different subtypes that can bind to the CCR5 co-receptor, and this is ones that do are known as the R5 HIV. Whereas there are X4 HIV versions, or a different subtype, that bind a CXCR4 tropic. Not really stuff you need to know, but thought it was interesting. So in order to use therapy, we need to define what type of HIV subtype it is. If it is a CCR5 tropic HIV, then you may proceed to use Maraviroc or this entry inhibitor in your patient. However, if a patient comes back and their subtype is either CXCR4 or dual tropic, then it's less likely that this agent is effective in a person with HIV because then we're not having that interaction with the CCR5. Okay, moving on from Maraviroc or our entry inhibitor. Next we have our fusion inhibitor. The mechanism of action of this medication is it actually binds to the GP41 here. So that's a glycoprotein that's located on the HIV envelope instead. A subunit of the viral envelope there. And then it prevents the conformational changes required for the fusion of the virus to enter into the host cell. So essentially the drug will block GP41 and then the interaction between GP41 and co-receptors down here do not allow for fusion. So on the next slide here, I just have a little summary of each of the medications. So for the entry inhibitor, we have Maraviroc, or brand name Cellzentry. How it works there, so reviewing it, binds to the CCR5 receptor of the CD4 cell and prevents the GP120 receptor from binding and entering into the host cell. Those are some side effects of the medication, and those are the potential mutations that can occur. So Maraviroc is actually an oral tablet that's administered, and it's generally well tolerated. I don't see very many patients on it. I think I know maybe one, but it's very, very uncommonly used, and so I don't have too much clinical experience with it. And then next we have the fusion inhibitor, or called enfuvirtide, or brand name Fusion. How it works is it binds to the GP, that should say 41 subunit of the viral envelope, and prevents the conformational change required for fusion to occur. So Fusion is actually, instead of an oral tablet, it is a subcutaneous injection, which makes it not very fun. A lot of people don't really like to receive shots if they don't have to. So another thing to think about when placing patients on therapy too is whether or not they're able to give themselves shots, and whether or not they can tolerate it, really, and be adherent to that, right? Okay, of course, to keep you all engaged, I have some few pop quiz questions. However, I guess it doesn't really work when you're doing a recording, but we'll go ahead and go through them anyway. So question number 1 I have is, which of the following is required prior to starting a patient on Maraviroc? So as you remember, Maraviroc is the entry inhibitor. So that will require a specific type of testing that will determine a specific type of co-receptor. And if you can recall from 2 slides ago, that the answer is actually B. It's the tropism testing to determine what HIV subtype the patient has, to see if that HIV will be interacting with CCR5 or CXCR4 co-receptor for binding. Okay, next we'll get into kind of our first class of medications that are most commonly used, which are the nucleoside reverse transcriptase inhibitors. I will refer to them as NRTIs for short. So these are the agents that are recommended as the backbone of an HIV regimen. So if you can recall from our previous video, 3 active ingredients are required to make a complete ARV regimen. Right? So 2 out of the 3 are going to be 2 different NRTIs. So since NRTIs and our next class of medications, NNRTIs, so that's 2 N's in the beginning instead of 1, they involve the enzyme reverse transcriptase. So I'm actually going to take a little bit of time to talk about the enzyme with you so you guys can understand kind of where the drug does its magic. So HIV reverse transcriptase is an enzyme that is involved in the critical HIV reverse transcriptase process. So as you can recall here, looking back at that photo, I know I've cropped much of it out, but binding and fusion are up here, right? So we'll move down to the next step, which is where reverse transcriptase does its thing to change HIV RNA into HIV DNA. So one HIV virion, fun fact, contains approximately 50 reverse transcriptase enzymes. HIV reverse transcriptase process is a multi-step complicated process that results in a copy of linear double-stranded HIV DNA to be generated. So that's essentially what's happening here. We'll get HIV RNA to generate DNA. So NRTIs, or nucleoside and nucleotide reverse transcriptase inhibitors, were the first agents that were available for the treatment of HIV. Although they're less potent against HIV than NNRTIs are, and PIs are, and integrase inhibitors are, they have a central role in antiretroviral treatment, as we can recall, and they remain a part of current standard of care. Actually, kind of touching base on what we talked about earlier too, PrEP, or pre-exposure prophylaxis, the medication that'll help decrease your chance of transmitting HIV in HIV negative individuals, or acquiring HIV in HIV negative individuals, actually has 2 NRTIs as a full kind of treatment or prophylaxis regimen for preventing HIV. So how NRTIs work is they actually competitively inhibit HIV reverse transcriptase. So when there's competitive inhibition, we're talking about there's something else that's also trying to bind there, right? And so, but it doesn't inhibit host DNA polymerase. So that's the nice thing about NRTIs is they're selective for reverse transcriptase. And they're not going to go and freely start, messing up or mucking up any of the host genetic material or host DNA polymerase at all. So these guys are going to be competing and binding against human nucleotides that are freely floating around. And once they do compete and outbeat regular nucleotides, they will bind into the chain as essentially a nucleotide and incorporate itself, causing premature chain termination. Um, Interestingly, once you guys see the structure that I have for these agents on the next slide, you'll kind of maybe get a better understanding of how they work and why they work the way they do. So, long story short, they compete, they bind into where the next nucleotide would bind under DNA um, transcription here, and they essentially stop the transcription from happening. So here's an example of an actual nucleotide. So if you can recall, we have 4 nucleotides: adenosine, guanosine, cytosine, and thymidine. And so structures of nucleoside or nucleotide reverse transcriptase inhibitors essentially mimic what a nucleotide looks like. The only difference is, as you'll see here on AZT or zidovudine, that first approved medication for HIV. And here this is an image of, I want to say, back of you. And so it's missing a substituted 3' hydroxyl group. So therefore you can't get chain elongation, right? Because the next sugar will attach here. And so without that hydroxy available at the 3' position, you cannot get chain elongation, and therefore it terminates the entire process of transcription. Here are some examples of currently approved NRTIs. We have a couple different agents here: abacavir, tenofovir, emtricitabine, lamivudine, and zidovudine. So a lot of these agents are kind of co-formulated, which means that they are made together because they're always essentially going to be given with another NRTI, right? Because just to reiterate, the 3 active ingredients in an ARV are going to be 2 NRTIs and either one of the other classes of medications that we briefly kind of went over. So PIs, NNRTIs, or integrase inhibitors are going to be the third active agent. And so we have a Truvada, which is approved for PrEP. So this is going to contain tenofovir and emtricitabine. As you'll see, maybe throughout this lecture too, I'll have kind of the names of HIV medications, and some of them help to give away certain things. The last letter in their 3-letter abbreviation will tell you what nucleotide analog they are. So lamivudine and emtricitabine both have NZ, and so they'll be a cytosine analog. So their structure is most likely going to look like what cytosine will look like. Side effects include mitochondrial toxicity. However, the newer agents are very well tolerated. Emtricitabine, tenofovir, especially the tenofovir alafenamide formulation of tenofovir, are generally well tolerated. And then mutations here. So discrimination, reverse transcriptase essentially avoids binding the NRTI, so it's able to distinguish that, hey, this is kind of a bad guy. I shouldn't be binding him. Or there's primer unblocking, which is removal of the chain terminating NRTI from the terminus. So there's actual mechanisms of excision of the NRTI. And then once it's excised out, transcription can proceed as regular. Agents, so a lot of the times we'll see these NRTIs co-formulated with another NRTI, right? Because they're always used as two in a backbone. So Truvada has two different components, and same with Epsigom. And so it helps to decrease the amount of pills that a patient is taking, because if you have to be on two NRTIs anyway, you might as well be taking them as one tablet a day. Will help with adherence. Okay, so that kind of wraps up the NRTI portion. So we'll move on to NNRTIs, but before we do that, I have a second kind of pop quiz question to go over with you. Which of the following best describes the structure of NRTIs? As if I didn't talk about that enough. So between all of the agents, if you can remember correctly, an NRTI has a specific kind of substitution at the 3' position that does not allow for chain elongation, and that essentially ceases transcription. So that's going to be the 3' substitution. That's not a hydroxyl group. So the answer here is C. The hydroxyl group will actually proceed to allow for chain elongation. And so next I'll start talking about the protease inhibitors, or the PIs as we call them. Um, as you can recall, protease, the enzyme itself, is responsible for cleaving enzymes to allow for activation of the mature virus. So here's kind of a photo of where it cleaves. So HIV protease cleaves at all these sites here and leads to all these um, active enzymes. So these encode for either protease inhibitor, other protease inhibitor, reverse transcriptase, and other enzymes that are involved in HIV replication so that they can be active to go on to infect another CD4 cell. So essentially how the drugs work, is that they competitively inhibit protease inhibitor by resembling amino acids unique to the viral protease. So this prevents processing of viral proteins into functional proteins.